PCR. These are what's called 7 transmembrane receptors, also known as serpentine structure. The reason for that nomenclature I'll show you in just a minute. The prototype of the G protein coupled receptor is rhodopsin, and I'll show you a little bit of the history of that in a minute. I'll define for you what I mean by 7 transmembrane segments. I'll show you that the N-terminal region of the receptor is extracellular and the C-terminal region is intracellular, and both of those have very significant implications in terms of the structure and function of these proteins. So rhodopsin. All right, so rhodopsin is the light receptor in the rod outer segment. This is so the function of rhodopsin is to bind to a photon of light. So this is a unique receptor. The agonist for this receptor is light. When rhodopsin is exposed to light, it becomes bleached, and it then undergoes a conformational change to produce signal transduction through a G protein, which we talked about last time, called transducin, or G sub T. Rhodopsin comes from the rod outer segment, which is a very specialized tissue or organ, very, very well studied, particularly in the middle part of the 20th century. Very easy to obtain this tissue. You would go to a slaughterhouse, get some cow eyes, and take the retina and just shake it, and the rod outer segments will just drop out into your little dish. And it turns out that the rod outer segment is extremely specialized. It's just a series of membranes that contain virtually nothing else but rhodopsin and its signal transduction molecules. So it's not like a cell like we think of, like a nerve cell, which does so many different things. So the rod outer segment is so specialized that we call it like a bag of rhodopsin. So it was really easy to take these rod outer segments and purify the protein rhodopsin using techniques in the mid-20th century that we would consider primitive today, certainly long before the molecular biology techniques we use today to sequence DNA and RNA and so forth to obtain the structures of all the proteins we do today. So this protein was purified by classic biochemical protein purification techniques. A lot of work, a lot of years it took to do this. And so when they purified this protein rhodopsin, they learned that it was a single subunit protein and that it contained 7 transmembrane segments. So for those of you who don't know exactly what that is, if you look at a membrane protein, just looking at the amino acid structure, or any protein actually, there's a random association of charged and uncharged amino acids in a normal protein, particularly in a cytoplasmic protein. But in a membrane protein, the amino acids are not randomly associated. And what you'll see if you look at the entire amino acid sequence of a membrane protein, you'll see sequences of 20 to 25 amino acids that are all uncharged. Okay, so you're talking about leucine, isoleucine, phenylalanine, tryptophan, but you won't see glutamate, aspartate, lysine, things like this. And so what people have learned from this is that these are transmembrane segments, 20 to 25 amino acids of uncharged amino acids that sit in the membrane, allow the protein to locate itself within the lipid bilayer. And so when they looked at the structure of rhodopsin, they found 7 of these things. Now, if you have an odd number of transmembrane segments, that means that one side of the protein is going to be facing inward and the other side is going to be facing outward. That's just the way it has to happen. If you have an even number, you're going to have both sides either inward or outward. So if you have 7, you know that one is out and one is in. So they did a number of experiments, and they learned from this that the C-terminal end of the rhodopsin molecule was facing inside the cell, and the N-terminal end was facing outside. So this was the only receptor of this particular type that had been known for many, many years, and there was no reason to suspect that rhodopsin was really a prototype of any kind kind of superfamily of receptors. In those days, receptors were defined by their pharmacological characteristics, not by their signal transduction characteristics. So this one bound to light, the beta-adrenergic receptor bound to norepinephrine, acetylcholine, so forth and so forth. So no one considered that there would be anything common between rhodopsin and any other receptor because it bound to light. But then people began to study other G protein coupled receptors, and they found that they all look the same, all right? So whether the receptor bound to light, this one is an opiate receptor, the beta-adrenergic receptor, the alpha-2 adrenergic receptor, they all follow the same structural rules. And that's why we call them superfamilies. And these superfamilies are all related according to their signal transduction properties. So these are all G protein coupled receptors, and they all work by the same rules, even if they work on different G proteins. So here is an example of a mu-opioid receptor. This is a receptor that is coupled to GI and GO, and once again, you can see these 7 transmembrane segments. So we define them as TM1, TM2, 3, 4, 5, 6, 7. There are 3 extracellular loops 1, 2, and 3. There are 3 intracellular loops, okay? There's an N-terminal extracellular portion of the receptor that once again faces outside the cell, and there's a C-terminal portion of the receptor that faces inside. I'll come back to this structure more in just a minute, but I think you can clearly, it doesn't take a structural specialist to figure out that the ligand binding site must be associated somewhere up here, because the ligand, of course, is going to be your agonist or antagonist that's either being released from other cells or be it administered in vivo. So clearly, somewhere in here, either in the N-terminal extracellular phase or in these extracellular loops, you're going to have a binding site for the ligand. And it also doesn't take a genius to figure out that inside the cell, you're going to have the signal transduction properties of the receptor, because that's what the business end of the receptor is built to do, okay? It's built to couple to the alpha subunit of the G protein and induce those changes in G protein structure and function that lead to the ultimate biological response. So once again, we can, without knowing anything, we can assume that G protein alpha subunit will interact somewhere either in the 3 intracellular loops or in the C-terminal region. Where are you?
Is it always that a GPCR's N-terminus is extracellular and always the C-terminus is intracellular?
As far as I know, I don't know any of them that are different than that. It almost has to be. The N-terminal region is not critical for function very much, but the C-terminal is absolutely the business end of the molecule for for the function. So I can't imagine how it would work any other way. For many years, the only thing that we knew about the actual 3-dimensional structure of the receptor was based on rhodopsin, because rhodopsin was the only protein that people could isolate in sufficient quantity so that they could get X-ray crystallography. You may or may not know that in order to get a full 3-dimensional structure of a protein, you have to have an X-ray crystallography analysis done. And in order to do that, you have to crystallize the protein. If you have a soluble protein, a protein from the cytoplasm, crystallizing the protein is not so hard to do. But if it's a membrane protein, it's very difficult, because because of the transmembrane segments, these are very lipophilic molecules. They don't crystallize well, and it illustrates what's going on. What's really happening is that the receptor is coiled in a very specific way, and here are the 7 transmembrane regions of the receptor of rhodopsin, which are all coiled in a very specific way to allow the receptor to have its function. And what they learned from this is a couple of things. Number one is that primarily the ligand binding, particularly the agonist binding, occurs in the regions around TM1 and TM2. So here's TM1 and TM2, and most receptors, not all, but most receptors have their ligand binding site right in here. Originally, people thought that it would make sense for the ligand to bind in this big, long, N-terminal tail here, but that is not the case. I don't know of a receptor where the ligand binding occurs up here at all. So the ligand binding occurs right here, and it's in the membrane interface. So when an agonist is binding to the receptor, it's not just floating out here, it's actually entering into the interface between the lipid bilayer and the external milieu. Okay. The C-terminal region is the region that binds to the G protein. So the G protein alpha subunit binds in this region here between the third intracellular loop and the C-terminal intracellular tail. These P's refer to phosphorylation sites. Those are very important that I'll talk about a little bit later on. So this is where the alpha subunit binds, but which alpha subunit binds, okay? So remember, we have subfamilies of these receptors so the beta adrenergic receptor binds to GS, the alpha 2 adrenergic receptor binds to GI and GO, the alpha 1 adrenergic receptor binds to GQ. So norepinephrine binding to 3 different subtypes of the adrenergic receptor, activating 3 different types of G alpha. So you have subfamilies of receptors whose homologies really occur right down here in the C-terminal or intracellular region. So this is what I mean by this. You have subfamilies of intracellular regions. So when you sequence a new GPCR, you can tell immediately whether that receptor is going to be a GS coupled or a GI coupled, because you can do the amino acid sequencing down here, and all of these things are related. All right, so all the receptors that are GS linked have similar homologies in this area, okay, because they're binding to the alpha subunit of GS. Whereas if it's a GI linked receptor, all of these areas will be in common for GI and GO. So what I mean by this statement is that the G protein homologies are determined by the intracellular regions, and the pharmacology, which is the specificity of the receptor, are determined by the recognition site. So once again, what's the difference between an alpha 2 and a beta receptor? Well, the pharmacology is the same. So the alpha 2 receptor and the beta receptor are fairly similar up here because they're both binding to norepinephrine. However, if you look at alpha 2 receptors and muscarinic receptors, which are both GI coupled receptors, those two receptors would be very different up here because one is binding to acetylcholine, the other binding to norepinephrine. However, their structures down here would be similar because they're both binding to GI. So all you have to do with all this is just remember the recognition, the binding, the pharmacology of the ligand binding, the specificity, all occur here in the transmembrane segments. The function, the actual identity of the effector that's produced by this receptor, determined by the intracellular areas. Using a series of extremely clever genetic techniques, heroic kinds of things, Brian Kobilka won the Nobel Prize just a few years ago on determining the X-ray structure of these proteins. So now we understand what they look like, and it really is an elegant series of stuff. So G protein coupled receptors come in all different kinds of flavors. You have subfamilies, you have monoamine receptors, nucleosides like adenosine, acetylcholine, lipids, which would include cannabinoid receptors, and peptide receptors. And so they all have slightly different structures. I don't have time to go into this, but the structures of these things are pretty well worked out. We know pretty much which amino acid, for example, is responsible for binding to a particular portion of the structure of norepinephrine and epinephrine. And so the reason why these kinds of studies are so important is it allows pharmaceutical companies and other people to do what's called in silico binding analysis, okay? So you can actually computer model drug design now using the precise structure of these agonist binding sites, you can design drugs on a computer that should be not only very potent at these sites, but very specific and really distinguish between multiple receptor subtypes. So now we've really entered into the 21st century in terms of drug design, where we don't even, we not only do we not need animal behavioral stuff anymore, but in some cases we don't even have to start out with random screening techniques. So the whole function of what's going on here is that the receptor changes its conformation when it binds to agonist. And let me bring back my two-dimensional cartoon. This is not very sophisticated, but it's much more informative, I think. So if you can imagine the agonist is binding to this site here, and it induces a conformational change which allows the G protein alpha subunit to dissociate away from this receptor. And remember, that was the portion of the G protein cycle that we called activation. Antagonists bind in very different ways, and antagonists don't bind right here, for example. Antagonists bind at different sites along this receptor, and when they do bind to that site, they do not induce that conformational change, but by doing so, they prevent the binding of the agonist. So people have mapped these things out pretty well. It turns out that remember the G protein cycle, and okay. So here's the G protein cycle, and remember that in the basal state, the receptor exists complex with the G protein heterotrimer, and in that state, it is a high affinity for agonist. And then when the agonist binds to the receptor, everything dissociates, and the receptor becomes a low affinity state. So you have these two affinity states, which depend upon the ability of the receptor to bind to the G protein alpha subunit and heterotrimer. So once again, high affinity agonist binding, the receptor is coupled with G alpha beta gamma, and once the agonist is bound, the G protein dissociates and you have low affinity. Now you can induce these low affinity states by throwing in a high concentrations of GDP or non-hydrolyzable GDP analogs. So once again, and this goes back to a discussion we had last week with receptors and different states, you can throw in a non-hydrolyzable GDP analog like GDP gamma S, and you can actually allow this cycle to go in the absence of agonist. It's called a constitutive cycle. And when you do that with a non-hydrolyzable analog of GDP, the GDP gamma S stays on board, and you induce the formation of a low affinity state. So that's what I was talking about last week when I said that you could distinguish between high affinity and low affinity states with different guanine nucleotides. So here, remember, we had this. This is a different version of a slide I showed you last week. But here in the basal state, you have a mixture of high and low affinity states. This would be the high affinity state where the receptor is coupled to the G protein, low affinity state where it's dissociated. When you throw in a guanine nucleotide like GTP gamma S, you shift all of these receptors into the low affinity state. Notice that not only are you shifting this curve to the right, but you're changing the slope, and you should understand that by now, okay? Because here you have a low slope indicating a combination of two states, and a higher slope, a slope of 1 really, indicating only one state. And we talked about this last week, and again, this is now you understand. This is exactly the slide I showed you last week. This combination of high and low affinity sites with the D2 receptors. You shift it with a guanine nucleotide into the low affinity state, and now you understand what's happening because you are basically using a guanine nucleotide to allow this cycle to move forward and shift the receptor. All right, so the standard model for a G protein coupled receptor is this: a single subunit receptor, 7 transmembrane segments, receptor binding, G protein activation. Everything looks very simple. But it turns out that some of these receptors exist as dimers, where you have two receptors interacting together. And the most dramatic example of that is this guy here, the GABA B receptor, okay? GABA is gamma aminobutyric acid. It is the predominant inhibitory neurotransmitter in the brain. When we think of GABA, we normally think of the GABA A receptor, which is an ionophoric receptor. That is the predominant inhibitory receptor in the brain, and it's the target of many of your anti-epileptic drugs and sedative, anti-anxiety drugs. But there's also a G protein coupled GABA receptor, okay? And it's the GABA B receptor, all right? And this is a receptor that's coupled to GI and GO. It's an inhibitory receptor, and it functions much like any other G protein coupled receptor. So some years ago, they learned that there were two subtypes of the GABA B receptor. There's a GABA B1 and there's a GABA B2. That's also not unusual. Many receptors exist in subtypes. So what they did is they cloned, they sequenced the gene for the GABA B1 receptor. And like you do in these kinds of experiments, once you have the gene for it, you transfect the gene into an acceptor cell of some kind so that you could study the receptor in detail. So they took this gene and they transfected it into the cell, and the receptor was expressed on the membrane, but it did nothing. Okay, the GABA B1 receptor expressed into an expression, any kind of expression system you want, did not function. It was totally inactive. So they didn't understand this. It made no sense. There, a lot of theories were going around. Another group sequenced the GABA B2 receptor subtype, and they did the same experiment. They transfected the receptor into a cell. The receptor was expressed. It went into the membrane, and that worked out very well, except for the fact that just like the GABA B1 receptor, the GABA B2 receptor didn't work. Either one of these receptors transfected into a cell by themselves didn't work. And I don't know who had the genius idea to figure out what to do, but another group decided to transfect both of these two subtypes together, and son of a gun, it worked. All right? So what they learned from this is that the GABA B1 receptor and the GABA B2 receptor dimerize, okay? They form a dimer. And this dimerization for the GABA B receptor is absolutely critical for its function. The GABA B receptor does not work unless these two receptors come together, okay? And when they do that, they produce different kinds of signaling mechanisms depending on how these two receptors are activated. And I'll go into the different types of signaling a little bit later on. By the way, before I forget, let's go back to my original cartoon. I did mention the ligand binding site is here in the transmembrane segments. The G protein site is here. What does this site do? So there's this big, long N-terminal region of the receptor, and it has these little yellow Y-looking things on it. This cartoon is supposed to illustrate gangliosides. So these gangliosides are not required for ligand binding because you can remove them and it doesn't really usually affect the ligand binding. But what they are required for is to properly insert the receptor into the membrane. So you have to remember how these receptors are formed. They're, the DNA and RNA for these receptors are formed way up in the nucleus, okay? And then the protein is translated up there by ribosomes. And then in a nerve cell, this protein has to be transported by microtubular elements all the way down the axon until it reaches the axonal area. And at that point, it has to be inserted properly into the membrane. And it can't just be inserted in a random way, okay? You've got to insert it in the right direction so that this portion of the receptor is facing outwards and this portion is facing inwards. So how does the cell know how to do this? Well, it's these gangliosides that allow you to do it, because these gangliosides bind to the inner surface of the cell membrane. And it's like a lock and key arrangement. So you can imagine this receptor being transported up inside the cell here, and it reaches the inside of the membrane. These gangliosides attach themselves to the inside surface of the membrane, and that not only allows the protein to be incorporated into the plasma membrane, but it allows the receptor to be incorporated in the proper direction, all right? And the reason I remember to talk about this now is because in this experiment where they were doing these two GABA B receptors, these proteins were being inserted properly. One of the big theories was, you know, maybe for some reason when we express either one of these two receptors in the cell, they're not being inserted in the membrane properly. Maybe there's not the right ganglioside present. Maybe the cell doesn't have the right machinery to make the ganglioside correctly, and the receptors just aren't going in correctly, and they're all messed up in the membrane. But that turned out not to be the case, because they're inserted properly, absolutely well. It's just that they require these two proteins to work. Question?
Does the receptor have to bind to both of those to have signal transduction effect, or does only one of those in the dimerization have to be?
Does the receptor have to bind? You mean does the
So
agonist have to bind?
Sorry, the ligand.
Yeah, okay, so that's a really good question. And the answer is no, okay? The ligand can bind to one or the other or to both, and there are various kinds of complicated possibilities that determine all kinds of different signaling pathways here. So it's a complicated process, but I'll talk more about different signaling in just a few minutes. So this is called a heterodimer, okay? Because these are two different receptors. Even though they are subtypes of the same receptors, they are considered to be different receptors because they are products of two different genes, all right? So there's but there's also homodimerization. So for example, you could have two beta-2 adrenergic receptors coming together. And here is obviously you don't need to know any of this, but this is just a list of some of the receptors that look like they homodimerize. So getting back to your question a minute ago, it seems like there are pharmacological differences. There are some cells in which the single subunit receptor is the predominant one, and that has one type of pharmacology, and there are other types of cells in which the homodimer is more predominant, and that changes the pharmacology as well. So this is a brand new area. It's an area that we don't know very much about, but it obviously increases the diversity of the receptors that are involved, because now we don't simply have one gene producing one receptor, which is what we always thought was the case. We now understand that one gene can produce multiple types of receptors depending on how it dimerizes. And this is heterodimers. Look at all the different types of heterodimers that you can have. Some of them are closely related, like the beta adrenergic, beta-2 and the alpha-2 adrenergic. But some of them aren't related much at all, like for example adenosine and dopamine. So once again, this is a fairly new area. Yeah, if you're working in the area of receptors, you'll certainly see a lot more of this kind of stuff in the future. I want to spend a lot of time now talking about mechanisms of desensitization. Remember last week I talked about the desensitization of the nicotinic ionophoric receptor, and we spent a little bit of time showing you how different affinity states changed the desensitization. The desensitization with G protein coupled receptors is much different and involves a lot of different steps, and so we're going to go through this in detail. First thing I need to do is make you aware that there are two different types of desensitization, one called homologous and one called heterologous. And they're mediated by different effects. This kind of desensitization has been known for many, many years, and it's based on the following type of experiment. Imagine you have in your hands two different cell types, and these cell types contain two different G protein coupled receptors, beta adrenergic and prostaglandin receptors. So this is an experiment done many, many years ago. Both of these are GS-coupled receptors. They both stimulate cyclic AMP. One cell undergoes homologous desensitization. So what we mean by that is you treat that cell with chronically with norepinephrine, let's say for a period of 24 hours. And when you do that, you get a desensitization of the beta adrenergic response, and exactly the same the thinking behind that is what we talked about last week. Once again, receptors like to maintain a homeostatic state, so that when they're overstimulated, they're going to go into a desensitized state. So the norepinephrine receptor, the beta receptor, becomes desensitized, and now when you add norepinephrine to these cells, you don't get any stimulation anymore, okay? Because the receptors have lost their ability to produce a response. That's the definition of desensitization. But it's specific, it's homologous because it's specific to the beta receptor, all right? Because if you stimulate these cells with prostaglandin, you do get stimulation of cyclic AMP because the prostaglandin receptor has not been desensitized by the chronic treatment with norepinephrine. That should make sense to you, okay? It's a very desensitization, a specific phenomenon specific to the receptor that's being overstimulated. But there's also another type of desensitization called heterologous, where all different types of receptors are being desensitized. And here is another cell, once again, chronically stimulated with norepinephrine. Once again, you lose the beta adrenergic response as you would predict. But as you might not have predicted, in these cells, you also lose stimulation by other receptors. So you also get desensitization of the prostaglandin receptor as well. So I'm going to show you how both of these things work. We understand very precisely now exactly the kinds of biochemical mechanisms that mediate both of these phenomena. So this is once again a summary slide. I'm going to go through this slide quickly, and then we'll go through the different steps in detail. G-protein coupled receptor desensitization is a multi-step process. The first steps are quite reversible. They are designed to be very reversible as receptors are stimulated for a short period of time, chronic, overstimulated for a short period of time. But then they become irreversible if the receptors are stimulated, overstimulated for a longer period of time. So in the reversible steps, we have phosphorylation of the receptor on the C-terminal region. This leads to uncoupling of the receptor from the G protein, loss of the G protein, and effector activation. So if a receptor is stimulated overstimulated for a short time, these are the steps that occur. But if it's stimulated for a long time, then you get sequestration of the receptor into the cell. Then that brings about a loss of membrane receptor binding sites, which is downregulation, and degradation of the receptor by intracellular protease enzymes. All right, so let's go back to our simple two-dimensional cartoon and see how this works. So normally, the whole system is designed to work very quickly, not as quickly as on an ionophoric receptor, but still relatively quickly. So the agonist binds to the receptor, okay, inducing a conformational change, releasing the G protein, and as soon as that happens, the receptor shifts into a low affinity state and the agonist dissociates, right? That much is clear. So that G protein cycle intends to go very fast. But suppose you overstimulate the system so that as soon as the agonist dissociates, you've got so much agonist on board that the agonist can bind immediately back on again. In other words, the agonist can even bind to a low affinity state because there's so much agonist around. The longer the G protein receptor stays in the low affinity state, the longer this portion of the molecule becomes exposed. Now remember, in the basal state, this internal portion of the receptor is hidden by the G protein itself. When the G protein dissociates, now this portion of the molecule contains these phosphorylation sites that become accessible for specific kinases. These are enzymes that phosphorylate amino acids on proteins. The longer this portion of the receptor stays open and accessible, the greater the probability is that these kinases will phosphorylate this portion, the C-terminal portion of the receptor. Once the C-terminal portion of the receptor is phosphorylated, the G protein can no longer bind, okay? So it can no longer work. And so this receptor becomes dead, all right? Because it simply exists in the low affinity state, it's phosphorylated, it's uncoupled from its G protein, so no matter how much agonist binds to the receptor in this state, it won't work because there's no G protein present to do this. So this is the desensitized state where the receptor C-terminal region is phosphorylated and the G-alpha subunit dissociates. Now, what are the enzymes that do this? The enzyme originally was called BARK, beta-adrenergic receptor kinase, because it was identified by Robert Lefkowitz, who won the Nobel Prize down the road at Duke, and he works on beta receptors. But very quickly, people learned that it wasn't specific to beta receptors, that it occurs for all receptors. So it's now called GERK, G protein receptor kinase. And there's all kinds of them around. I forget, I'll talk more about that in a minute. So some of these residues, some of these amino acid residues here are phosphorylated by GERK, and that produces desensitization. But there are other phosphorylation sites that are not phosphorylated by GERK, and they're instead phosphorylated by other kinases like protein kinase A. Protein kinase A is cyclic AMP dependent protein kinase. Okay, so when you have a GS coupled receptor, you increase cyclic AMP. The cyclic AMP stimulates protein kinase A, and then protein kinase A phosphorylates all kinds of, like hundreds of receptors in this, hundreds of proteins in the cell. So the difference between these two enzymes represents the difference between homologous and heterologous desensitization. If a receptor is phosphorylated by GERK, it's a homologous desensitization because this GERK is specific for the particular receptor that's being overstimulated. So if this is a beta-adrenergic receptor and it's being overstimulated by norepinephrine, GERK will phosphorylate the beta-adrenergic receptor, but it can't phosphorylate a prostaglandin receptor because the prostaglandin receptor is not being desensitized. So this is homologous. However, if a receptor could also be phosphorylated by protein kinase A. So remember, if you're overstimulating the beta-adrenergic receptor in the cell, it's producing an increase in protein kinase A. The protein kinase A could then phosphorylate another receptor, and that's exactly what you get in this particular scenario, okay? So you're treating chronically with norepinephrine. The GERK phosphorylates the beta-adrenergic receptor, and that desensitizes the beta receptor, but the protein kinase A stimulation can phosphorylate the prostaglandin receptor, and that produces heterologous desensitization. So there's lots of phosphorylations that are going around. Now, this is a totally reversible step because as soon as these amino acid residues are phosphorylated by GERK or pKa, they can also be dephosphorylated by other enzymes, phosphatases. Okay, a phosphatase is an enzyme that removes phosphate from amino acids. So, if there's a lot of phosphatase activity around, the phosphatase can cleave the phosphate from this amino acid here, and once the amino acid becomes dephosphorylated, then the G protein can come back onto the receptor, and the receptor can regenerate its high affinity state and now become active again. So the dynamics of this process, this balance between kinase and phosphatase activity, depends upon how much agonist is involved. If you continually stimulate with agonist, the kinases will predominate. Phosphorylation will predominate. Desensitization will predominate. But if you remove the agonist from the receptor environment, then the phosphatases will take over, and the receptor will be regenerated back into its active state, and now you have a fully active receptor. So that's why we say that this particular phase of the receptor desensitization is reversible, okay? Very easily reversible. And so you can imagine that this kind of process occurs all the time, and it does. You can actually measure minute-to-minute effects going on here. You can see phosphorylation and dephosphorylation receptors going on in the cell, in the intact cell, over a period of minutes. So it's very common, very, very much a part of the process. But if you keep stimulating the receptor for a long period of time, hours, days, months, whatever, then you start to get into another phase of this phenomenon where the cell says, okay, all right, well, I had this great idea to desensitize, but it's not really working so much because you're still stimulating, so I'm going to just get rid of these receptors. And this is how it works, all right? So you've got this phosphorylation and internalization phenomenon. So in this first part of the cartoon, you've got the receptor being stimulated by the agonist. That's the agonist up here. And you've got this GERK, okay, that's phosphorylating the receptor. Now, if the receptor stays phosphorylated for only a short period of time, then it can reverse itself. But if it stays phosphorylated for a long period of time, it becomes accessible to another protein inside the cell known as beta-arrestin. Beta-arrestin is a signal for internalization. Once beta-arrestin binds to the, now remember, beta-arrestin cannot bind to the dephosphorylated receptor, all right? So the receptor has to be phosphorylated in order for beta-arrestin binding to occur. But once beta-arrestin binding occurs, then this complex of the receptor and beta-arrestin can then interact with the internal membrane and form what's called a clathrin-coated pit, all right? And this is the first step of endocytosis, wherein the receptor, the phosphorylated receptor bound to the beta-arrestin, undergoes an endocytosis phenomenon where the clathrin-coated pit is sequestered inside the cell. And once this thing is sequestered inside the cell, then you have proteolytic attack that will hydrolyze this receptor, and then now the receptor has been removed from the membrane, and now you get downregulation or a decrease in the number of receptors. Okay? So this part becomes irreversible. Okay? Irreversible being a relative term. Now, I don't mean irreversible meaning that they'll never come back, but what it means is that in order to get active receptors again, the cell will now have to synthesize new protein. And it'll take a while to do that, because you've got to start in the nucleus by increasing RNA synthesis, then translating the RNA into protein, and that protein has now got to be transported down the axon, all the way down through microtubular transport, down to the axon, insert the protein into the membrane into the right way. So it takes a while to do that. So I guess you could call it pseudo-irreversible. All right. So I need you to remember this phenomenon of beta-arrestin binding to the phosphorylated state of the receptor. And this is the endocytosis mechanism. Downregulate first. So first we have desensitization, then we have downregulation. But beta-arrestin does something else too. And I'm not going to spend a lot of time on this because Dr. Hallett will, I think she'll talk more about it when she speaks to you next week. This is a cartoon which illustrates receptor trafficking. And there's a lot of things going on here. So once again, the first step of desensitization you know about already. Okay, you've got this. This is the first step here. They've listed as a heterologous desensitization, which is PK mediated. There's a GERK mediated phosphorylation. Now there's a beta-arrestin binding, but there's actually three things that can happen once the beta-arrestin binds. I've told you about one of them already, okay? So one thing that can happen is that the beta-arrestin complex receptor can be endocytosed inside the cell, and it can be hydrolyzed, all right? So this is signal 3. That is the hydrolysis of the receptor and the ultimate downregulation phenomenon. So that's one thing that can happen. But something else can happen too. The receptor can be trafficked, recycled back into the membrane, all right? So and every cell is different in this, and every receptor is different. So some cells have a very rapid endocytosis and receptor degradation phase, so this degradation part is predominating. However, other cells and other receptors, the degradation phase is much slower, and if the degradation phase is slower, then this endocytotic clathrin-coated pit can actually be recycled back into the membrane. And so the receptor can actually be trafficked back in. And so once again, in the intact cell, you can actually see this over a minute-to-minute phase. You can see internalization occurring, and the internalization either being terminated by degradation or being re-trafficked back into the membrane. So each cell, each receptor has its own recipe on how it likes to do this. Some goes the degradation route, other goes the trafficking route. It really depends on whether the cell wants to regain that receptor or not. Some cells would just as soon degrade the receptor. Other cells would like to bring the receptor back and allow the whole process to occur again. So those are the two of the signals that occur when beta-arrestin is on board. But there is a third signal that can occur as well. And that is that once beta-arrestin binds, the beta-arrestin coupled receptor can actually create another biological signal, all right? So beta-arrestin dependent signaling is different from G-protein dependent signaling. G-protein, we've talked about this already. You've got cyclic AMP, you've got ion channels, a bunch of other things going on. But there are other pathways which don't depend on G proteins themselves. They depend on beta-arrestin. So just imagine that during this period of time, between the time that the receptor becomes phosphorylated and bound to beta-arrestin and the time that the receptor either traffics back or degrades, between those two events, there is a signaling that occurs. And so in other words, there's an activation of a variety of different kinases in the cell that are beta-arrestin dependent. So when we think about today, when we think about GPCRs, we think about G-protein dependent signaling, and we think about beta-arrestin dependent signaling. And why is this important? Because the biological consequences of those two pathways are completely different, okay? The G-protein pathway is protein kinase A, cyclic AMP. It phosphorylates one set of proteins in the cells. But the beta-arrestin dependent signaling involves an entirely different group of intracellular enzymes. Enzymes that you're going to learn about next week, like GERK and ERK and all kinds of these other things. And they are phosphorylating an entirely different set of proteins inside the cell. So these are brand new concepts, but now we're thinking about biased and unbiased agonists as potential drug targets. A biased agonist is an agonist that would work through one of these two signaling pathways and not the other, okay? So a biased agonist might work through G-protein but not beta-arrestin, or the other way around, might work through beta-arrestin and not G-protein. An unbiased agonist works through all of these things equally well. So think about the concept of developing drugs now. We're not just thinking let's design a new drug that works at the beta adrenergic receptor. We're not thinking that way anymore. Let's think about designing drugs that are biased or unbiased. And if they're biased, which pathway do we want to go with, okay? Do we want to go the beta-arrestin route, or do we want to go the G protein route? Maybe the G protein route produces the good effects of the drug, and the beta arrestin route produces negative side effects. Maybe we could design better drugs if we could focus on one pathway as opposed to another. So keep this in your memory when Dr. Hallett speaks to you next week, when she talks about all of these different enzymes. We've talked about protein kinase A and GERK here today, but she's going to introduce a lot of these other enzymes, the ERKs and GSKs and things like that. They're all going to be enzymes that are important in other biased kinds of pathways. Question?
If you want to prevent the internalization of the receptors by blocking the beta arrestin, would you have to take into consideration what would happen by also blocking the signaling that the beta arrestin dependent would do?
You're absolutely right. Yes. So if you could block the beta arrestin to block the internalization, but you would also block that pathway. And maybe that's a good thing. Maybe the beta arrestin pathway is not one you want to have, so maybe that would be good. But on the other hand, maybe the beta arrestin pathway is really good. And like I said, some cells don't do this degradation very much. Okay, so some cells have a lot of internalization, a lot of degradation. Other cells don't. So each cell is different. Each receptor is different in the way they balance signaling versus degradation. There's no set rule that applies to all of them. Well, as usual, there are all kinds of complexities. There are all kinds of GERKs involved. 7 GERKs, I see, and at least 2 different forms of beta arrestin. Each of these have different they do the same thing. Okay, they all phosphorylate receptors, and they all bind to receptors that are phosphorylated. But they have different distributions in brain, and they have different specificities. There's a lot of research being done now on trying to figure out the specificities of these different GERK enzymes and different arrestins. We don't have time today to talk about that, but it's an important field. Okay, so before I finish off in this, let me summarize the whole issue with desensitization, and let's think about the contrast between G protein coupled receptors and the nicotinic receptors we talked about. Remember, the nicotinic receptor became desensitized when it entered into a high affinity state. Okay, and the whole reason for that is that in the high affinity state, the acetylcholine remains bound and it doesn't come off, and the receptor just sits there. The G protein coupled receptors are exactly the opposite. Okay, they are in a high affinity state in their active form, in their basal state. And then when they become desensitized, they switch over into their low affinity state. So the processes that are involved in these different types of receptors, the time course that occurs, time course is slower here for the GPCRs versus the ionophoric receptors. All of them are quite different. But both of them use this concept of the relationship between affinity and dissociation. This theme comes up over and over again. The way that they use it varies depending upon the family of receptor that you're looking at, but nonetheless, in every case, you're looking at high affinity states with slow dissociation versus low affinity states and rapid dissociation. So just keep that in mind as you think about these things, and it'll hopefully make sense. Now, over the next 15 minutes or so, I'll talk about the G protein coupled effectors. This is very, very brief summary. I just don't have time to go in detail, and I know that Dr. Hallett will talk some more in terms of the kinase activities that are associated with these. So there are 1, 2, 3, 4 different G protein coupled effectors, and we'll talk about them individually. Let's talk about the easiest one first, the cyclic GMP phosphodiesterase, which is the effector for light transduction in the rod outer segment. So this is the signal transduction mechanism that's connected with rhodopsin in the rod outer segment. Once again, the agonist being light. Cyclic GMP phosphodiesterase is an enzyme. And once again, let me just mention all of these are the effectors. These are the effectors that are either activated or inhibited by activated G proteins. So the G protein cycle you already know about. We know the agonist binds the receptor, produces the activated G protein. Remember that there are two messengers for the activated G protein. The activated G alpha, which is bound to GTP, and the free beta gamma. So you have both of these messengers going out to either activate or inhibit effectors. And some of these effectors will be activated by beta gamma, others will be activated by the activated G alpha. All right, so back to rod outer segment. Here's the enzyme. And this enzyme takes guanosine triphosphate, GTP, and it changes it into cyclic GMP. I'm sorry, so there are two enzymes here. Let me back up. There's an enzyme called guanylyl cyclase, which takes GTP and converts it to cyclic GMP. That's done in the basal state. But our signaling enzyme is this one, phosphodiesterase. And phosphodiesterase basically converts cyclic GMP into 5' GMP. So this is the enzyme that is important in the rod outer segment. I've always thought this is a fascinating process. It's not my area, so I don't know a lot about it, but it's still very interesting how this works. So in the rod outer segment, you're targeting cyclic GMP-gated potassium channels. All right, so normally in the dark, GTP is being converted into cyclic GMP, and the cyclic GMP produces a dark inward potassium current. All right. So in the dark, cyclic GMP activates an inward potassium current that brings potassium inside the rod outer segment. When rhodopsin is activated by light, and that process is called bleaching, this enzyme is activated. Cyclic GMP is hydrolyzed to 5' GMP. In the absence of cyclic GMP, the inward potassium channel is closed, and the dark current is halted. And when the dark current is halted, that produces depolarization of the neurons in the eye and leads into an active action potential into the visual processing centers. All right, so the dark current is an inhibitory current which prevents cell firing. Once you get rid of that inhibitory current, the cells can fire and you see. All right. Now, one of the things that's nice about this process is that they've done a really nice mathematical calculation of the amplification process that's involved. So remember I told you last week that receptors are amplifiers, okay? That one molecule of agonist binding to a receptor produces massive changes inside the cell because you're getting all of these different steps that are occurring, okay? And every G protein coupled receptor does this in their own particular way. This one is nice to talk about because they've really calculated the math involved here. So, in general, it's been shown that the activation or bleaching of one rhodopsin receptor leads to over 100,000 molecules of cyclic GMP hydrolyzed per second. Okay, so and this is how the math works to get this. So one rhodopsin activates several hundred proteins of transducin. So when you activate rhodopsin, it's not just hitting one G alpha T. It's activating a lot of them at the same time. And each molecule of transducin activates one molecule of phosphodiesterase effector. And each molecule of phosphodiesterase hydrolyzes more than 1,000 molecules of cyclic GMP per second. So the rod outer segment, which is responsible for your night vision, okay, black and white vision, is incredibly sensitive, okay? If you're totally dark adapted, you lock yourself up in a dark closet for an hour or two, you can in fact perceive 1 photon of light. And that's because of this system, okay? This system is so remarkably sensitive because the receptor signal transduction system is amplified at each step along the way. And it's not just true for this system, but the other systems as well. But this is a great place to talk about it. All right, so that's transducin and rhodopsin. The second effector that I'll talk about is adenylyl cyclase. Of course, this is the historically the first of all of the signal transduction systems to be identified by Earl Sutherland back in the 1950s. And the enzyme adenylyl cyclase basically takes ATP, adenosine triphosphate, and converts it into cyclic AMP. Remember that the target of cyclic AMP is cyclic AMP dependent protein kinase, otherwise known as protein kinase A. And what PKA does is to phosphorylate specific serine and threonine series residues in protein, and there are dozens of proteins within the cell that are phosphorylated by PKA, and that leads to the function of this receptor. But getting back to the effector, adenylyl cyclase is a very large intrinsic membrane protein. So the molecular weight is over 100,000 kilodaltons, or 100 kilodaltons. And its structure resembles that of a transporter protein. So there's a lot of interesting evolutionary history about adenylyl cyclase, because a protein that resembles adenylyl cyclase is actually seen in extremely primitive bacteria. And the function of this protein is it's an adenosine or ATP transporter protein. And so the bacteria transports ATP inside the cell, because obviously ATP is a food product for that particular bacterium. And so this is a food protein for the bacterium. But interestingly enough, it's also a sensory protein, because bacteria will move according to the gradient of where the food is, okay? So if there's ATP out there, the bacterium will move towards it. And it's this protein that allows that to occur, all right? Because this is the protein that's allowing the uptake of adenosine inside the cell. And the reason I mention all of this is because I view adenylyl cyclase as a primitive sensory protein. Even in primitive bacteria, it's actually performing a signal transduction role, even though it's primarily a role of producing solutes, food for the cell. Nonetheless, it allows a primitive sensory response. So in my view, signal transduction goes all the way back to bacteria. Now, adenylyl cyclase is a complicated series of proteins. There are at least 7 subtypes of these enzymes, and each of them have different distributions in brain, and they're very different in terms of what they're activated by. So some of them are purely activated by calcium and calmodulin. Those are the calcium calmodulin dependent cyclases. But you also have G protein dependent cyclases. Some of them are stimulated by GS alpha, and some of them are stimulated by beta gamma. And each of them have different distributions and slightly different kinetics and so forth. So we don't know exactly how this thing works in terms of why one cell will have one type versus another, but certainly it's a complex effector system. So cyclic AMP is not only involved in producing a number of changes inside the cell, but cyclic AMP is also involved in the third of our effectors, the ion channels. There are 2 principal ion channels that are coupled to G proteins. One is calcium, and the other one is potassium. And there are 2 ways in which ion channels can be activated by G proteins. One is by phosphorylation, and one is by the G protein itself. So this slide tells you about the mechanisms of how the ion channel is activated by phosphorylation. So basically, the great example of this I'll talk about in a minute is the calcium channel in the cardiac myocyte. If a channel is phosphorylated, it can induce a change in the ion conductance. And if it's a calcium channel, phosphorylation of the calcium channel opens the calcium channel and allows calcium to go in the cell. And there are two ways that you can phosphorylate a calcium channel through a G protein. You can do it through GS, okay? GS activates protein kinase A, and that phosphorylates the calcium channel. Or GP or GQ activates protein kinase C. Both of these are kinases that stimulate calcium channels. I'll talk about PKC in a minute. This is a relatively slow activation. So it takes greater than 300 milliseconds to open a calcium channel by phosphorylation. That doesn't sound like much, but it's actually pretty slow. And the reason why it's slow is because you have to go through all these steps. The agonist binding the receptor dissociating from the G protein activating cyclase forming cyclic AMP phosphorylating, activating PKA activating the ion channel. 5 or 6 steps involved in that, so it takes a while. Here's a great example of how this works in the heart. So you know that epinephrine, or norepinephrine, stimulates heart rate, stimulates contraction of heart muscle. It does this by increasing calcium influx into the muscle cell. Calcium is required for muscle contraction. You should know that. Hopefully you do. So here's how this works. The epinephrine binds to the beta-adrenergic receptors. Beta receptors are coupled to GS. The beta receptors increase cyclic AMP inside the cardiac myocyte. Cyclic AMP stimulates protein kinase A. Protein kinase A phosphorylates a T-type calcium channel and increases calcium conductance. Okay? So basically, binding of epinephrine to the beta-adrenergic receptor phosphorylates the calcium channel, increasing calcium uptake into the cell. Now let's talk about the other type of ion channel, which is those that are directly coupled to G proteins. And these would be primarily the potassium channels, or the inhibitory channels. So in this case, you're not doing all of these steps, okay? There's no cyclic AMP involved, there's no PKA, there's no phosphorylation. You're simply activating or affecting the channel directly through the activated G protein, either the activated G alpha or the beta gamma. So it's a very fast response because it doesn't require all of these second messengers. So once again, this is a response that's quite a bit faster, less than 300 milliseconds. The mechanism here is really simple, okay? The agonist binds to the receptor, it activates the G protein. You can have either free, the GTP bound G alpha or the free beta gamma to activate the ion channel directly, all right? This mechanism is the mechanism of action of GI and GO, okay? All right, so GI and GO acting on ion channels in a relatively fast way through this mechanism. Forget this thing about GTP gamma S. I'm not going to talk about that. So let's go back to the heart cell, okay, and see how this works. So remember that in the sympathetic autonomic system, the sympathetic neurotransmitter is epinephrine. It contracts the muscle, the heart rate. The parasympathetic system is acetylcholine, reduces heart rate. Okay, acetylcholine reduces muscle contraction in the cardiac myocyte. And it doesn't do this through nicotinic receptors. Nicotinic receptors have nothing to do with this. This parasympathetic autonomic response of acetylcholine is strictly due to muscarinic G protein coupled receptors, and specifically involving the subtype of muscarinic receptors that are coupled to GI and GO. So here's how it works. Acetylcholine binding to the M2 muscarinic receptors. That produces an activation of GI-GO. the GI-GO dissociate, either the beta gamma or the free GI subunit activates the potassium channel. This produces hyperpolarization, so that reduces cell firing. And when that happens, that reduces muscle contraction. So, so that is the fast response to this whole process. There is also a second phase of acetylcholine effects in the cardiac myocyte that's slower, and that works through phosphorylation. So the M2 receptors not only act directly on the potassium channels, okay, through this direct mechanism, but they can also affect calcium by inhibiting cyclic AMP. Remember that GI and GO inhibit adenylyl cyclase, inhibiting cyclic AMP, inhibiting protein kinase A, and when it does that, an inhibition of cyclic AMP, that will then dephosphorylate the calcium channels and reduce calcium conductance. So there's actually, the cholinergic muscarinic receptor actually has two different mechanisms of action, one fast and one slow. Final effector I'll talk about is the effector that's coupled to GP or GQ, depending on the method, on the nomenclature you prefer. And the effector is generically referred to as phosphoinositol turnover. Phosphoinositol, phosphatidylinositol, is an intrinsic lipid that's present in every lipid bilayer in every cell. So it's a ubiquitous lipid. But there's a specific enzyme that's activated by G proteins called phospholipase C. And what phospholipase C does is it converts phosphatidylinositol into two different molecules, diacylglycerol and IP3, which is inositol triphosphate. These are two different messengers, okay? These produce two entirely different mechanisms. Diacylglycerol stimulates another protein kinase in the cell known as protein kinase C. And protein kinase C is even more active than protein kinase A, phosphorylates hundreds of proteins in the cell to increase protein cellular activity. IP3, inositol triphosphate, on the other hand, increases intracellular calcium. So what IP3 does is it binds to the endoplasmic reticulum. Endoplasmic reticulum, one of the functions of that in the cell is to store calcium. So intracellular levels of calcium in the cytoplasm are normally very, very low because the endoplasmic reticulum takes all the calcium that's been taken up from outside the cell and it stores it, okay, inside that organelle. So it's inactive. But what IP3 does is it binds to a specific receptor on the endoplasmic reticulum and it releases the calcium from that storage compartment. So now you have a big increase in intracellular calcium, and the calcium can then go on to do a variety of things like increase neurotransmitter release and produce muscle contraction. So once again, two entirely different signals produced by one enzyme, all right? So GQ or GP is a stimulatory G protein because it's increasing protein kinase C activity, that increases the activity of the cell, and it also increases the cytoplasmic concentrations of calcium, and that produces cellular activity as well. So now you can see how this mixture of stimulatory and inhibitory G proteins produces their effects through these different effectors. And once again, I think you'll see when Dr. Hallett comes on board next week, how these effectors can then go down the signal transduction pathway and activate a variety of other kinases that ultimately mediate the final biological response. All right, questions about any of this? So I want to, if I can get this to work, I want to spend 10 minutes or so talking about the homework. Hard with, there you go. As I said earlier, I really I really think, I consider this sort of problem a learning mechanism, so if you turn something in, you'll get all kinds of partial credit. What I'll do is I'll post this answer, or I'll have someone post this answer onto Canvas so you can see what's going on. So here's a restatement of the problem. Remember, we had diaphragm tissue that was lesioned, and we assayed tritiated nicotine binding in both the control and the lesion muscles. So the first thing you need to do is to convert the Curies into DPMs, and then convert the DPMs into concentrations, in this case nanomolar. This is not difficult to do, but once again, if you've never done it before, then it can be confusing. So the goal of this, and on the table, I provided you with the units that you need, nanomolar, and I told you that the assay volume is 1 mil. So nanomolar is nanomoles per liter, or picomoles per mil. So we're really trying to solve for picomoles. That's what we're trying to get to. So 25 curies per millimole is 25 millicuries per micromole, 25 microcuries per nanomole, or 25 nanocuries per picomole. So this is the range we want to look at. So the question is, how many DPMs are in a nanocurie? Because if we can solve for nanocuries, we can get a picomole out of that. So there's nothing complicated about this. A curie is 2 times 10 to the 12th DPMs. A millicurie is 10 to the 9th. Microcurie is 10 to the 6th. A nanocurie is 2 times 10 to the 3rd. Well now, gosh, I don't need exponents anymore, okay? 2 times 10 to the 3rd is just 2,000 DPM. That's easy. So 25 nanocuries is 1 picomole, so that means that 50,000 DPMs is 1 picomole. So all you need to do here is to divide the DPMs by 50,000, and it's done. The whole problem is done. So you go into Excel, you take these things, you divide everything by 50,000, and you convert the DPMs into nanomolar, and that's what you get. So here it is, nanomolar added, specific DPMs bound, all in nanomolar. I'm sorry, here's the DPMs bound, the nanomolar bound, okay? To get free, you simply subtract the bound from the total, and you get that. And then you do bound over free, and you get that. Notice that the ratio of bound over free is always less than 0.1, so it's all in zone A. Remember, we said that receptor binding should always be less than 10% bound over free. So this is fine. You do the same thing for the lesion. And there it is. So all you have to do is plot it, and that's what you get. So scatter plots are linear, and you don't need a supercomputer to look at this thing and figure out that you have a parallel shift in the 2 scattered plots. Okay, so that should indicate to you immediately that there's no change in the KD, that there is a change in the Bmax, and in fact, in the lesion case, we're getting an increase in the Bmax, and from the scattered formula, you know that the KD is the negative reciprocal of the slope, and the Bmax is the x-intercept. So it's really simple to go ahead and use those plots to solve for the KD and the Bmax. You get the KD from the slope, you get the Bmax from the intercept, which is from the y equals mx plus b, and you come up with that. All right. So notice the control KD is 0.59, the lesion KD is 0.61. I tried to make these come out as close as possible. So from my point of view, they are the same. Notice the Bmax changes from 0.015 nanomolar to 0.41 nanomolar. So it's almost a 3-fold increase in the number. Yes.
You used 2 times 10 to the 12. So is it okay if we use the 2.2 and we just got slightly different?
That is not a problem. You'll get full credit for that if you did that. That's not a problem.
Can you back up, please, to the previous slide? The one after that. No, the graphs of the scatter plots. How did you, when you were graphing this, I'm confused how you got that I got completely different equations.
Slopes?
on my lines.
Okay,
Yeah,
all
my
right.
slopes are
So
completely
now obviously I did this on Excel or something or other that calculated slope for me, but you can do it by hand, okay? Rise over run, you can do that. You can-
I mean, Excel
Yeah.
did it for me.
Yeah,
but
okay,
I got
all right.
way different ones, and I was,
Well, you-
my table was completely the same up here, so I was really confused how you jumped from the table to this and got something.
I'll have to take a look at your data and
All right.
see what happened, and we'll go from there. And I have been known to make mistakes myself, so that's always, I reserve the right to make mistakes. So, here it is. No change in the KD, increase in the Bmax. What is going on here? Hello? This is denervation supersensitivity. Just exactly what we talked about in class. Very simple. We said in class that when you remove the neurotransmitter from the environment, the postsynaptic cell or the muscle responds by increasing the number of receptors and not increasing the affinity. So this is a great example of denervation supersensitivity that occurs over a long period of time after the presynaptic nerve cells have been removed. The other thing I said in class, which I doubt that many of you picked up on, is supersensitivity is a great example of spare receptors. So the secret to question 3 is that you now have a spare receptor issue, okay? The cells have responded by increasing the number of receptors. So now you can do exactly the same thing we did in class we had this problem in class where we had a 10-fold excess of spare receptors. Now you just have to plug in numbers that are different from that, but the calculation, the process is exactly the same. Supersensitivity is an example of receptor reserve. Use the Langmuir equation to calculate the effects of receptor reserve on the affinity of acetylcholine. All right. So the lesion increased the number of receptors by a factor of 2.7. So this is a receptor reserve of 2.7. That's basically the difference between 0.015 and 0.41. Now, how do you get from that to receptor occupancy? If you go back to our lecture notes, remember I said that those cells with receptor reserve had a 10-fold excess of receptors. So all you had to do is occupy 10%. 10-fold excess needs full response. All you need to do is occupy 10%. So it's really simple. All you have to do is do the reciprocal of the receptor reserve. So 1 divided by 2.7 means that all you need is 37% receptor occupancy to produce full response. So now you have everything you need. The EC50 of acetylcholine would occur at half of that, which is 18% occupancy. And so you just plug it into the Langmuir equation, solve for D, and now you see that in the lesion tissue, the EC50 for acetylcholine is now 0.29 micromolar instead of being 1.3. So this is just another example of how increasing the number of receptors can increase the sensitivity of the system to the agonist. Okay. I doubt that many of you got this, but so that's
Very accurate.
exactly. So but that's okay. All right. You know, like I say, I'm not here trying to pass you and fail you on these concepts, but if you, there's nothing complicated about it. It's complicated because you haven't seen it before, okay? But now that you go through this thing, it's not rocket science. It's really a very natural sort of mathematical consequences of what happens when you increase the receptors. Question?
Where are you getting the 18.5? That's
Yeah.
Okay, so once again, the receptor reserve is 2.7. So I calculate from the reciprocal of that 37% occupying the receptor would produce full response. And that goes back to what I said in class. Remember I said if we had a 10-fold excess of receptors, you only need 10% to get full response. So 100% response, you occupy 37%. So, but we're trying to solve for the EC50. The EC50 is the concentration of acetylcholine that produces 50% of the maximum response. So, acetylcholine occupying 37% would produce full response. So you just take half of the full response. And the EC50 of acetylcholine would occupy only 18.5% of the receptors, because it's half of 37. Yeah?
I have a question. So you said before that because there was an increase in Vmax but no increase in affinity, it
Yep.
was showing the desensitization. But now for the next question, you're saying there's an increase in affinity.
Yeah. Yes,
So
yes,
how are
yes.
they going
That's a
to-
really good question. So she's saying here you don't see any change in affinity, but now when you look at the agonist, you do see a change in it, and that's the trick of supersensitivity and receptor reserve. So in this particular scenario, we're just looking at the ability of tritiated nicotine to bind to these receptors. There's no function involved in the binding assay here. All we're looking at is the affinity and the number of receptors for tritiated nicotine. But in the next part of the problem, we're not talking about nicotine anymore. We're talking about the agonist, acetylcholine. And what we're saying is that if you increase the number of receptors, and each one of those receptors have the same affinity, but by increasing the number, you increase the affinity of the agonist for the receptor. And that's why receptor reserve exists. That's why the cells do it, because the more receptors you have, the lower the concentration of agonist is going to be. And I like when I talk about receptor reserve, I like to use a target practice analogy. I don't know if you guys talked about this in class or not, but if you set up a bunch of tin cans on a fence and shoot them with a BB gun, if you only have 3 or 4 cans on the fence, you're not likely to hit anything. Okay, I'm not likely to hit anything. But if you put 150 cans on the fence, you're likely to, even if you're looking the other way, you're likely to hit something. Well, that's exactly what's happening here. Okay, the cell is increasing the number of receptors, and by doing so, it increases the probability that the agonist will make a hit. Okay, and so it's target practice. That's all it is. Okay. Okay. So, like I said, if you certainly, I don't expect many of you to get question 3, but hopefully you learned something from this.